Hey everyone, and welcome to The Explainer. Today, we're diving into something way more thrilling than just your standard textbook process of cell division. We're going to explore meiosis, which is honestly the ultimate biological casino shuffle. It's this incredibly precise, high-stakes architectural process that guarantees the absolute uniqueness of every single individual and ultimately ensures the survival of entire species. Literally, without it, you wouldn't be you. Okay, let's dive into this. Here's our roadmap for today. First, we've got the blueprint of diversity, followed by the Great Shuffle, the final split, the engines of variation, and finally, we'll wrap up with errors and their real-world impacts.
Starting right off with Section 1, the blueprint of diversity, setting the stage for the ultimate shuffle.
So think about it like this. If our reproductive cells carried our full set of DNA, combining them would endlessly double our chromosomes every single generation. We'd have a massive genetic overload. To prevent that, nature uses meiosis. It's this highly specialized cell division that reduces the chromosome number by exactly half, taking a diploid cell and producing haploid gametes, things like sperm, eggs, pollen, or ovules. It is absolutely the ultimate reduction strategy.
All right, moving right into Section 2, Meiosis 1, the Great Shuffle. Let's look at this first round of division.
This first round is known as the reductional division, and it's where the aggressive genetic shuffling actually kicks off. Prophase 1 is arguably the most crucial stage here, and it's broken down into 5 distinct acts. Just think of the chromosomes like characters preparing to trade secrets. In leptotene, they condense and make their appearance. In zygotene, matching homologous chromosomes from each parent find each other and pair up. That's called synapsis. Then, pocketene brings the actual crossing over of material. In diplotene, they start pulling apart but hold on tight at specific connection points called chiasmata. And finally, diakinesis clears the stage, with the chromosomes fully condensed and the nuclear membrane breaking down.
Now, what's really interesting about this slide is that key event happening right in the middle during pachytene, crossing over. This is the main event, guys. Non-sister chromatids literally trade pieces of their DNA. This isn't just some minor detail. It is nature actively rewriting the genetic script to create entirely new allele combinations. It's a real game changer.
After prophase 1, the cell hits the gas pedal through the rest of the first division. In metaphase 1, those paired homologous chromosomes align right at the equator of the cell. And get this, their orientation here is completely random. Next up, in anaphase 1, the homologous chromosomes are pulled apart. But notice this, the sister chromatids fiercely hold on to each other. They absolutely do not separate yet. Finally, in telophase 1 and cytokinesis, the cell divides, forming 2 haploid cells. The chromosome count has been halved, sure, but each chromosome still consists of those 2 tightly bound sister chromatids.
Which brings us nicely to Section 3, Meiosis 2, the final split. This is all about locking in those unique gametes. Now this round is an equistorial division, acting very much like standard mitosis. Let's rapid-fire through these final steps. In prophase 2, the chromosomes condense again, and new spindle fibers form inside our 2 new cells. In metaphase 2, the chromosomes line up at the cell's equator, single file. Then, in anaphase 2, the big moment finally arrives. Those fiercely attached sister chromatids are torn apart and pulled to the opposite poles of the cell. Wrapping the whole shebang up, telophase 2 and cytokinesis divide the cells one last time.
So let's look at the big picture and see how this resolves the entire 2-stage architectural process. We started with just a single diploid cell, our 2N base. And the grand finale? 4 completely distinct, genetically unique haploid cells. Pretty amazing, right?
That takes us to Section 4, the engines of variation, or how nature shuffles the deck.
Meiosis doesn't just cut chromosome numbers in half. It's practically a factory for genetic variation, creating the differences that make every individual totally unique. We can actually map this out as 3 distinct shuffles. Shuffle number 1 is crossing over, which as we just saw happens during prophase I. The result? Recombinant chromosomes that physically mix traits from both parents onto a single strand of DNA. Shuffle number 2 is independent assortment. This hits during metaphase I, when chromosome pairs line up randomly. This dictates exactly which combo of maternal and paternal chromosomes ends up in a given gamete. And finally, shuffle number 3 happens entirely post-meiosis, random fertilization. Because literally any unique sperm has the potential to fertilize any unique egg, the sheer mathematical diversity here is amplified exponentially.
And this brilliantly illustrates why all of this microscopic shuffling actually matters to the world at large. Genetic variation isn't just some cool cellular parlor trick.
It drastically increases the survival chances of entire populations. Because individuals have different genetic makeups, some are naturally going to be better suited to survive new diseases, climate shifts, or other crazy environmental changes. It provides the essential raw material for natural selection to work on, completely reducing a species' overall risk of extinction. This variation is for sure the very engine of evolution.
Let's pivot to Section 5, Errors and Real-World Impact. What happens when the machinery fails? Because, let's be real, with millions of cellular components moving, pairing up, and pulling apart, imperfections are absolutely inevitable. So what actually happens when this incredibly precise division fails?
Well, when the division fails, we generally see something called non-disjunction. This is the microscopic failure of chromosomes to separate properly during anaphase, either in meiosis I or meiosis C. Instead of one chromosome going neatly to each new cell, both might get yanked into one, leaving the other cell with absolutely none. This completely throws off that strict chromosome count we established earlier.
So, the crucial point here is that these microscopic separation failures aren't just invisible errors, they manifest as major real-world genetic disorders. When a gamete with an abnormal number of chromosomes is involved in fertilization, it leads to conditions like Down syndrome, Turner syndrome, or Klinefelter syndrome. So understanding the core mechanics of meiosis directly helps us trace the origins of these chromosomal abnormalities.
And that right there is why mastering this topic matters way beyond just passing a biology exam. Grasping the intricacies of meiosis, from its stunning precision to its potential flaws, provides a massive foundation for modern science. It directly influences how we study genetics, how we develop much more resilient crops in agriculture, how we approach medicine and fertility, and how we push the absolute boundaries of biotechnology.
I want to bring it all together with this really powerful thought from our source material. This variation is the foundation of evolution and adaptation. Think about that. Every unique physical trait, every innate resistance to disease, every tiny difference you see in a population around you, it all ultimately stems from this microscopic cellular shuffle. It's essentially the silent architect of life's resilience.
So, I'm going to leave you with this provocative question to chew on. Since meiosis ensures that every single gamete is a totally one-of-a-kind genetic blueprint, how might our deepening knowledge of this incredible process transform the future of medicine and the survival of vulnerable species? The blueprint is literally in our hands, and we're really just beginning to learn how to read it. Thanks so much for joining me on this explainer, and keep asking great questions.